So, good morning. My name is Rob Lavigne from the Lab of Gene Technology, and today I'm going to introduce the CRISPR-Cas system as a molecular scalpel. Actually, this is supposed to serve as an introduction to the other lectures as well, and what's really fascinating is how such a small enzyme can have such a huge impact on various fields. Obviously, today we'll have a lot of emphasis on agriculture, and if we go into this, and traditionally for over centuries, mankind has aspired to use domestications and breeding programs to improve the quality of the animals in agriculture. In this example is shown 3 chickens, all 56 years old, and you can see breeding programs from the 50s compared to 2005. You see that the animals of the same age have more than quadrupled in body mass. This using traditional breeding and so forth. But of course, this is taking a huge amount of time. So in the past, other strategies have been employed to improve this. So for example, this citrus has been exposed to radioactivity or to chemical compounds in order to induce mutations within that genome that result in desired traits, here a reduction in pits. But of course, you already feel that this is a very coarse way and potluck kind of approach where we don't understand the specific mutations that are causing these desirable traits. And therefore, it's very unspecific, unclean. Also, we don't know what other mutations may be introduced by such mutagenesis approaches. However, in the last 2 centuries, the genomic revolution has enabled us to sequence thousands of different organisms. And when we look back 20 years ago, the first human genome has been sequenced. But the cost of this genome sequencing has dramatically declined. You see here the straight line, Moore's law, which is the speed at which processors of a computer increase in speed exponentially. And you see that sequencing costs drop even further, even faster. And it's now possible to sequence a human genome for a few $100. In addition, sequencing technologies are miniaturizing in size to handheld devices, and this is resulting in new opportunities where now hundreds of thousands of human genomes are being studied into this genomic level to the greatest detail. That also means that we are now able much more to pinpoint within a genome where a specific error, what a specific position or site is that we wish to alternate and change. But of course, for this, we need molecular scalpels, which are going to specifically target the area we want to change. And the key ingredient there is to introduce a double-stranded break in the DNA molecule. In the next step, this is then repaired by the correct DNA, here highlighted in green, by non-homologous end joining or by homologous recombination, allowing you to delete, insert, and modify specific sites to do that. There are various biotechnological tools, so all of these scalpels have been developed in the last 20 years as well, including zinc finger nucleases, talents, and the focus of today, the CRISPR-Cas9 system. Yeah. Compared to the other two, you will see that CRISPR-Cas has some specific advantages. Yeah. So if we look in more detail into the region that we wish to modify, the only thing we need is a 3-nucleotide sequence called the protospacer adjacent motif or PAM sequence that needs to be located in the vicinity of the DNA molecule that we wish to change, here indicated in red. To this, we add 2 components. One component is the Cas9 enzyme, which is a nuclease enzyme that will cut the DNA, but it needs to be guided to the correct position. And for this, it uses the guide DNA, which is highlighted here. Yeah? Having a specific protospacer sequence that has been designed in such a way that it matches the target sequence that we want to alter. Yeah? The combination of this enzyme and guide RNA recognizes the PAM domain, and there will be a hybridization between the RNA molecule and the DNA molecule, which is opening up the DNA strand, and then the nuclease enzyme will cut the DNA at 2 positions, causing this double-stranded break. And this is the key technology that was made. The major breakthrough here is that we're using a very simple combination of a nuclease enzyme in combination with in total 100 nucleotide RNA molecule. Yeah. The 20 nucleotide sequence defines the specificity and ensures that a cut site will be triggered towards the specific position that you're anticipating. Of course, this has been gradually improved, and derived technologies have been made to further increase that specificity, but still, this makes Cas9 the important biotechnological tool that it is. The combination of this enzyme and this guide RNA that triggers the nuclease activity to the right position. So you may ask yourself, where is this coming from? What is actually the biology behind this CRISPR-Cas9 system? And actually, this is derived from bacteria. Bacteria which carry this molecule, Cas9 molecule, or other similar proteins that are present. But in this case, in the biology, we have a tracer RNA molecule and we have a protospacer RNA, the CRISPR RNA. And both combined are triggering mutations, are triggering cuts in the DNA. You may ask yourself, why do we want this cut reaction to occur? Well, actually, this system, the CRISPR-Cas9 system, serves as a bacterial immune system that protects the bacterial cell from foreign DNA. Furthermore, inside the bacteria, there is the genomic locus, as you can see here in the bottom, which encodes for the genes for Cas9 and so forth, and all of the other molecules that are needed, including the tracer RNA and also including the CRISPR RNA. And that's where we get the name, Clustered Regulatory Interspaced Short Palindromic Sequences, or CRISPR in short, is encoded on this genomic locus which many bacteria carry, and they themselves have an array of protospacer sequences that they have ready for the protection. You may ask yourself, from what does a bacteria need to protect itself? Well, from invading DNA, and this invading DNA is present for example, in viruses and plasmids. Yeah. These CRISPR sequences were first observed in the 1980s. In the years 2000, these have been identified as a bacterial immune system, and only in 2012 this has been converted into a biotechnological tool. But going back to the function, why does a bacteria need to defend itself? Well, among other things, bacteriophages. Bacteriophages are bacterial viruses that predate the bacteria. Yeah? These bacteriophages recognize the bacterial cell, inject their genetic material, and then convert this bacterial metabolism into a virus-producing machine, and subsequently amplify themselves and release the progeny phage while lysing the bacterial cell. A key step there is the injection of that DNA. It is this DNA that is going to be recognized by the CRISPR-Cas system, and in this way the CRISPR-Cas system serves as the immune system that I was talking about. Yeah. If it's able to block phage infection, in this way, it can save its own life, it saves the bacterial life. This is all quite complex, and you have to realize that within the bacteria, instead of in the biotechnological tool where we have the protospacer that we ourselves define, actually the bacteria has acquired over generations specific short sequences of different kinds of phages it has been exposed with. In rare events, those sequences become integrated into that CRISPR region and therefore offer immunity protection if progeny bacteria are infected by these bacteriophages. This is, of course, important, and it also illustrates the close interaction between bacteriophages and bacteria. Yeah. We talked about this host takeover mechanism, but you have to realize that the phages themselves are also evolving. And nice work by the Joe Bondi Denomi's group has shown that actually some phages even have the antidote against CRISPR. These anti-CRISPR proteins are functioning to block this defense mechanism. So this is sort of a co-evolutionary arms race, and it really goes into choosing the right weapons when you go to war. So this counterbalance between bacteriophages and bacteria is a really important aspect of biology, one that we study in great detail in our fundamental research, but all of this also has implications for phage therapy. Yeah? In our lab, we're looking to see if we can use bacteriophages to kill dangerous bacterial pathogens that are causing infections in humans, for example, infections associated with trauma implants and so forth. Yeah? And you have to realize that phages in that sense are quite unique because these phages are highly specific, recognizing a certain bacteria, and based on this, they infect the bacteria, lyse it, but in that process are becoming a self-amplifying antimicrobial. And that makes them of potential interest. But as I mentioned before, the intertwining of the relationship between bacteriophages and bacteria, which can carry this CRISPR-Cas system, is really important. Yeah? We have to know which phages are the right phages to use against certain bacterial species, against certain bacterial isolates. So in the lab, we can see this by these negative colonies where bacteria are not able to grow because they get lysed by the presence of phages. And sequencing hundreds of bacterial species allow us to identify which isolates have CRISPR-Cas systems, for example, or other defense mechanisms that are encoded on the bacterial genome. So we're using a lot of biology, a lot of molecular insights to ascertain that interaction between the two, but also we're using machine learning to discern the rules and the molecular determinants that are important when choosing the right phages. And this really goes towards applying a phage cocktail and designing a phage cocktail that's optimal for the use for certain patients. All of this is, of course, one aspect of introducing phage therapy to patients. There are legislative issues, there are production issues, there are processing issues, and of course the clinical implementation. And we're responsible for the molecular safety and the aspects that I described to you before. And this then results in phage cocktails that can be used. Yeah? The next slide is a bit more graphic, is an example of a patient which was transferred to the University Hospital in Leuven after 10 years of, let's say, bad treatment in other places. They were referred to the hospital. You see here implants that were had associated infections, and these were then through surgery. Then new repair was made and phages were administered. And this is one example of about 8 patients treated so far, which after this extended period of having infections associated with those implants have now been infection-free for 1 year. So I hope this shows you that CRISPR-Cas9 on one hand, is a very powerful biotechnological tool that enables an efficient and cost-efficient genome editing, there's also various derived technologies that not only introduce double-stranded breaks, but also other functionalities in terms of transcription and so forth. And this is possible in a broad range of species. I've also shown you that the CRISPR-Cas system is just one example of a diverse bacterial immune system that protects the bacteria against foreign DNA like bacteriophage predation. And this is, of course, one of the many factors that we have to take into account if we want to introduce phage therapy as a therapeutic complement to treat dangerous bacterial pathogens. Thank you for your attention.